Thank you to the organizer for inviting me to this meeting. I really appreciate this invitation. Today I will talk about stress. What are the effects of stress in the human brain? This is a very important, not only for the neuroscience, for the all people that study mental disorder, the origin of mental disorder. What are the effects of stress in the people that are exposed to high level of stress, for example, in the healthcare worker that's currently fighting with the COVID-19 pandemic, or some people that are exposed to a stressful experience? Why some people are more vulnerable or susceptible to this stress and other people are more resilient? Yeah? We know that in some people that are exposed to the stress, increase the prevalence of the development of several mental disorders, especially mood disorder, depression, anxiety, and post-traumatic stress disorder, and also some neurodegenerative disorder like Alzheimer's disease, dementia. There is a very important correlation with the stress experience in the life and to the development of Alzheimer's disease, for example. So first, what is stress? Stress is a concept proposed by the professor Hanselier last century. And this concept is related to our capacity of adaptation of our environment. Yeah. So how works this system in our brain? First, we perceive the stress from our environment by our sensory system that have a key role in the control of our stress response, because all of this information reach to the center of our brain in a brain area called amygdala complex or amygdala. And this area, and you can see here in red, is an area that control our emotional processing and also have a key role in the control of fear, yeah, and the stress response. There are different system circuit in the brain that control our stress response. For example, the endocannabinoid system, monoamine system, but the sympathetic nervous system and the hypothalamic pituitary adrenal axis, both have the most important role in the control of our stress response. A fast response is related to the sympathetic nervous system and the hypothalamic or HPA axis. Okay, first we perceive the stress from our environment and in the amygdala sense the danger of the stress information from our environment. Yeah, and you can see here, yeah, amygdala identify the danger, and then this information is sent to the hypothalamus. First, to prepare our body to fight with stress by the activation of the sympathetic nervous system and the release of adrenaline. And at the same time, but more slowly, is activate the HPA axis activity from the hypothalamus to produce stress hormone. But the most important stress hormone is released from the adrenal cortex, is the cortisol. Cortisol gives the energy to develop the behavioral response of the stress, the fight-flight response. So when we adapt to the stress from our environment, yeah, the technical name is eustress, our brain is connected with our environment, perceive correctly the signal from the stress from our environment, and our response are correctly development to adapt to our environment. But the problem is when we don't adapt to the stress that we feel. Yeah, for example, Now, during the COVID-19 pandemic, we work a lot in our house, but this hyperactivation of the HPA axis and the amygdala complex, specifically the excitability of the amygdala, affect other brain area very close to the amygdala, for example, the hippocampus, and affect our learning and memory process. And also, when the amygdala increase more their activation, the excitability specifically, affect our frontal cortex, and induce several morphologic chains that affect our emotional flexibility, learning flexibility, and affect our cognition, our decision-making, attention. So all of these alterations that induce this toxic stress increase the vulnerability or susceptibility to develop mental disorder. The most correlation of the stress is more to depression and anxiety disorder, and also Alzheimer. So what we know about this stress, the effect of the stress in the human brain. For example, here you can see two examples. The people that were exposed to the Japanese earthquakes in 2011, the effect of this stress in the human brain was correlated to hippocampal atrophy, and also atrophy in the orbital frontal cortex. And when this morphological alteration of the brain was more strong in the brain, that was correlated with the development of post-traumatic stress disorder. And another example is related to the stress induced by the terrorist attack in the state during the 2001. The people that feel this stress, the amygdala was atrophied. The gray matter volume decreased, but the excitability, the activity of the amygdala increased. At molecular level and cellular level, we know that this stress, when we don't adapt to the stress that we feel, this is translated to a molecular alteration in this area of the limbic system, the amygdala and the hippocampus, and also in the frontal cortex. This condition, this stress, increase, affect the expression of the glucocorticoid receptor and the cortisol release. And the imbalance in this system is translated in neuroinflammation, specifically in this brain area of the limbic system. And this is the first cellular alteration that correlate or associate the effect of the stress with the development of mental disorder, neuroinflammation. But we know that some people are very resistant or resilient to stress. When feel more stress, these people have the capacity to adapt very fast to this stress. The first investigator that focused his attention in this human capacity was Norman Garmency, and he studied how children adapt to stress. And Norman Garmency proposed this concept, resilient. Resilient is the ability to adapt to the adversity in a positive manner, but also more fast compared with other people. So what's happened in the brain of the resilient people? We know that that the people that develop active coping strategy with the stress, these people work more with the ventral medial prefrontal cortex compared with the people that are more vulnerable to stress, that develop a passive coping strategy with the stress, and also have the resilient people have more control of the sympathetic nervous system, and all different alteration or different change in the brain. But for study the neurobiology of the resilient to stress, it's necessary use animal model, yeah, and compare with the clinical study, yeah. The most common animal model in rodent for study the neurobiology of the resilient to stress is social defeat stress. That is correlate to the bullying in children or adults, in human. So some animals that are exposed to this behavioral paradigm are very vulnerable, very susceptible, yeah, develop depressive-like behavior, problem in memory, and also different morphological alteration in the brain, very similar to human condition, and other animals that are exposed to the same stress, yeah, are more resilient, yeah. So 2 years ago, I proposed this research question: how long does stress resilience last? Resilient is forever in humans. The people that born resilient to stress, okay, during all his life is resilient, or the resilient have a time? This is a very important question, because in an animal model for study the neurobiology of stress resilience is concentrate only when the end of the social defeat stress. My point of view is what's happened after, yeah? Okay, last June, we published this paper, and today I will only show some of these results. But if you want to check more information about this specific investigation, you can check the paper. First, we evaluate the effect of social defeat stress 1 week after end of this stress protocol. What's happened in the social behavior, because we can classify in rodent what animals are more resilient or susceptible to stress by the social behavior. Yeah, you can see the classical behavioral tasks to evaluate the social behavior in rodent. We use rats, male rat in our study. So we measure the time spent close to the interaction zone with other conspecific, other rats, the same sex and same characteristic of the experimental animal. And you can see here in the y-axis the time in the interaction zone. So some animals that are exposed to the social defeat stress 1 week after social defeat stress, the time in the social interaction zone was very similar to the control animal, no social defeat stress condition. But other group of animal, after 1 week of social defeat stress, decreased significantly the time spent in the social interaction task. And these animals here in red are susceptible to this stress, were susceptible to this stress, and here in blue, this animal who are resilient to this stress. So what's happening in the brain of this animal that were resilient and susceptible to stress? We evaluate the neuroinflammation in the hippocampus by the microglia activation and reactive astrocyte in this brain area. This was our first approach in this investigation. Currently, we are working what's happening in the amygdala and in the frontal cortex and other limbic areas. Yeah. So, okay, you can see here in this picture a representative picture of different experimental conditions. And here is the quantification of the microglia activation using the IGA1 antibody. And when you see more green, yeah, it's more neuroinflammation. Yeah, and you can see here, and after 1 week of social defeat stress, only susceptible animal show neuroinflammation in the hippocampus. But in the astrocyte, measured by the GFAP antibody, and you can see here that only in the susceptible animal, in the hippocampus of the susceptible animal, was more neuroinflammation compared with resilient and non-stressed animal. So what's happened in the strategy of coping with the stress? It is possible to study stress coping in animal model using the force swimming test. Yeah, when the animals are exposed to this behavioral task and develop more floating behavior, this is more correlation to a passive stress coping, and this is more correlated to the susceptibility or vulnerability to stress. When decrease the floating behavior, the animal show more an active stress coping, and this is more correlated to the resilience. And you can see here, after 1 week of social defeat stress, animals that were susceptible to social defeat stress had more floating behavior compared with the resilient animal. That means that in susceptible animal, the coping strategy was more related to a passive stress coping. Yeah, and using this other behavioral test, sucrose preference test, it's very simple. This behavioral test, the animal are exposed to 2 bottles, 1 bottle with water and other bottle with a solution of sucrose. And here in the y-axis, you can see the sucrose preference. And animals that were susceptible to stress decreased the sucrose preference compared with the resilient animal. This is more correlated to a depressive-like behavior. Yeah. So now, the most interesting result were what happened 2 weeks after social defeat stress. You can see here in the y-axis the time spent in the interaction zone, in blue resilient animal, and in red susceptible to stress animal, 1 week after social defeat stress and 2 weeks after social defeat stress. And you can see here the animals that were resilient to stress 1 week after social defeat stress lost their resilience after 2 weeks of social defeat stress. That means that in rats, resilient to stress, the time of the resilient to stress is 2 weeks. Yeah, the resilient to stress is not forever, it's lost. Yeah, and what's happening in the neuroinflammation? Yeah, here is the microglia activation, and you can see here in the hippocampus of the resilient to stress condition, increased neuroinflammation. A very similar situation here in the astrocyte activation. Yeah, increase the neuroinflammation compared with the control animal. Yeah, so what's happened at behavioral level? You can see here in blue that the animal that show after 1 week of social defeat stress a low level of floating behavior, yeah, increase the floating behavior 2 weeks after social defeat stress. That means that increase the passive stress coping with stress, and this is more correlation with susceptibility to stress. The same situation for the sucrose preference test. The animal that show resilient to stress 1 week after social defeat stress, that the sucrose preference was high, decreased 2 weeks after social defeat stress. Okay, the conclusion in rats, in male rats, is resilient to stress is not forever. We need to improve our resilience to stress. Yeah, because when improve our resilience to stress, decrease the possibility in human to developmental disorder related to stress. So now we are working in several experiments to find the molecular mechanism that is associated to the transition to the resilient to stress to vulnerability to stress, specifically the role of glucocorticoid receptor and mineralocorticoid receptor. We are using some drugs that are antagonists of glucocorticoid receptor and mineralocorticoid receptor. So in another experiment, we investigate what is the neural circuit related to stress resilience. What is the functional connectivity, the brain oscillation that communicate the information between different limbic areas that control our resilient capacity. The idea is use this functional connectivity, brain connectivity, to change to the stress vulnerability phenotype to a resilient phenotype. Another very important question that is associated to this result is what's happened in female rats. Yeah, there is the same situation to male rats. Yeah, the resilient, the time of the resilient is for 2 weeks. And another important question is, it is possible to improve our resilience to stress by a collaboration with a colleague in Liverpool, Francis McClone. 2 years ago, we proposed to use sensory system to improve our resilience to stress. The investigation of Francis is in human. He used social touch. He investigates what are the effects of social touch in the human brain. He found several years ago that in the skin, in the human skin, there are specific fibers, yeah, CT fiber. This is a technical name that is stimulated by soft stroking in the skin, yeah, and this is a physical stimulus in the skin, promote the activation of this nerve fiber, and this information reach to the insular cortex and induce the release of the oxytocin hormone. This is the most important hormone that is associated with our social behavior, promotes social behavior. Okay, Francis told me, okay, I think that this system could be related to the control our resilience to stress. Okay, we perform a simple experiment in rats using stroking at CLTM optimal velocity to activate this system in rat, and then expose this animal to a stress paradigm, chronic unpredictable mild stress, and evaluate the effect on stress response and stress resilience. Okay, I will show the most important result of this paper that was published last year in the European Journal of Neuroscience. Yeah. Here in the y-axis, you can see the corticosterol level. Corticosterol is the most important hormone, stress hormone in rodent. It's similar to cortisol in human. So here we can activate the HPA axis activity by the force wind, and then we can see the dynamic of the release of corticosterol in the blood. Yeah. So here in the white circle, you can see the no stress condition after activation of the HPA axis, increase the level of the corticosterol in the plasma, and then back to the base level. So in the black circle, you can see the stress condition, after activation of the HB axis activity, significant increase the level of corticosterol compared with the control condition. And this is the most interesting result. In blue circle is the stress plus 5 centimeters per second, the stimulation, stroking stimulation. And after activation of the HB axis activity, the level of corticosterol increased, but very comparable to the control condition, and compared with the animal that were stroked at 30 centimeters per second. So this result demonstrate that it is possible to control our stress response using a sensory system, the somatosensory system. And we are working in some experiment to use this idea in human, to prevent the development of some mental disorder, and also to use this technology in the treatment, for example, in depression, in other mental disorder that are associated with the stress. Finally, thank you to all the students that participate in our investigation in Valparaíso in Chile, and also the grant that support our investigation. Thank you for your attention.